This is the last in the last lecture in the series, based around Dick's book, and one that David was unable to make in the sequence, but has been kind enough to offer to provide it at the end of the sequence. As with many of the speakers, I've known David for quite some time, probably longer than just about anybody else who's been presenting. David did his Bachelor of Science in Physiology at the University of Western Australia, even though he's Irish by birth. And he did honours. Yes, he was transported like me. He did an honours with Brian Johnston, who many of you will know did some of the earlier and most seminal work on basal membrane and hair cell. And in fact, David then came to Oxford to do a DPhil, where I was a postdoc and a research fellow there. And I think he'd been working on cisplatin, which is inactivation of the outer hair cells, and looking at the impact that was having. David completed his DPhil at Oxford. Probably played a little bit of cricket while he was there as well, I think.
A lot.
And then had a series of postdocs. The most notable probably with Alan Palmer at Nottingham, the Institute for Hearing Research, the MRC Institute for Hearing Research. They did quite a lot of really nice work there together. Then he went on to become Professor of Auditory Neuroscience and Director of the Ear Institute that's at University College London. The Ear Institute has a long history in significant contributions to hearing. And then we managed to seduce him back. Well, I didn't, because I was overseas at the time, but others managed to seduce him back to Australia. And for nearly a decade, he's now been the academic director and professor at Macquarie University Hearing at the Australian Hearing Hub. So as a consequence, David's a key player in the Australian Future Hearing Initiative. So it gives me great pleasure to welcome my friend and colleague, David McAlpine, to take the floor.
Thank you very much, Simon. And I will now see if I can share the slides. Are they showing?
They are indeed, sir. You need to present. Excellent.
Good. Yes, so everything you've just heard about one ear on the other lectures, just add it to the other ear, and I think we're done, actually. That's pretty much binaural hearing, but of course it's not. And I want to give you a perspective today for binaural hearing based on Dick's book, which I also like from a historical perspective, because it does fit with some of the historical perspectives I've had from other sources. But also just try and broaden it out to thinking of how we create a sense of space, because we have got caught in the weeds for many years in binaural hearing, because it's a very important, very interesting perspective. Technically, there's a lot of interest in it. And it's got stuck in the psychoacoustic weeds for a long time and was a bit ghettoized from the rest of hearing. And yet we know that binaural hearing, or at least where things are in space, if you think of that as one element of binaural hearing, and where you are in space relative to a room, are fundamental canonical features of the three-dimensional world in a way that some of the communication things that we think about for hearing are not. And I think this plays out in terms of the fact that although we are vague about what our binaural hearing abilities do for us sometimes, compared with, say, pitch or common onset time or harmonicity, whatever, it's partly the only perspective on hearing that we are very sure about where the brain circuits lie and what they are doing. And I don't think that's an accident. I think that's because objects in the real world hold three-dimensional positions in all other sensory modalities, and you have to accept that. Whereas communication and abstract notions of voice or music, etc., are much more abstract and may not be found so easily in the brain circuits that we look for. So topics for discussion: what problems need to be solved by binaural hearing? A quick binaural history 101 from 150 or so years. Obviously, there are great characters in that. I won't talk about all of them. And then the duplex theory, this perspective that there are two mechanisms by which binaural two-eared hearing occurs: sensitivity to intraoral level differences and sensitivity to intraoral time differences. And some of the very basic psychoacoustics, I won't go into much detail about that. There is pretty much every JASA edition has multiple binaural psychoacoustics in it, generally from the same people and generally referencing themselves. So it's actually a big field in some ways, but actually surprisingly not well understood. I mean the field, not the psychoacoustics. We'll talk about some anatomy, physiology, and biophysics. And I think this is where I'd like to say there's been a transformation in the last 25 years. And I would also put myself square in the middle of that and say that we've probably transformed the way we think about binaural processing as we've got better technologies, as there's been better understanding of what neurons are doing, but also as we reframe the question away from signal processing to, for example, computational neuroscience. They're not separated, they are in the same space, but they have been explored in very different ways. And then I want to, at the end, talk a little bit briefly about pushing the envelope, the case for holistic spatial hearing, because as we've discussed, and I think as Dick made the point when we were first starting out in this venture about timing, and a lot of us get caught up in temporal fine structure or envelope or whatever. In fact, there's a holistic view of spatial hearing, which is really the whole signal, the fact there is an object out there and there are objects out there and worlds out there, and all of these things come in at once. We don't really have access to the individual cues in the way we think we do in the lab. The lab has been great for breaking those down, but putting them together again is the challenge, and I think that's something we need to discuss. So I always start with this with lectures. Where the fox that? It's important to remember that any sensory system we have that is highly evolved, and we have 10 millionths of a second discrimination performance for ITDs, evolved for a reason, evolved for survival. And the ability of this fox to locate mice a few feet below the snow and to survive is the basis for our senses. And in fact, we know that sensitivity to interval time differences, this 10 millionths of a second timing capacity that we have between the ears, evolved independently in birds, reptiles, and mammals after there'd been a movement from the aquatic to an air environment. So there are similarities in how that is done, but the important thing is that being able to get 10 to 20 millionths of a second resolution to be able to tell it 1 degree off to the side or the other challenges our understanding of the neural auditory code, because of course action potentials are a couple of orders of magnitude slower than that. So a 2 millisecond action potential turnaround time is much slower than your resolution for interval time difference. So it's always been of interest, and it's been of interest in a range of species, including owls, where people like Kanishi and Eric Knudsen have all provided great understanding or great interest in spatial hearing and spatial vision put together. So it's about survival. So it's not surprising that we've evolved very highly succinct and precise systems for locating where objects are or even hearing them out in the first place. Now we know the benefit for us is in that cocktail party problem. We know that one of the elements that helps us do this cocktail party listening is the spatial disparity between sources of noise and voices that we're trying to understand. We know that if you separate the angles of the noise from the talker, for example, you get a benefit, and there's binaural masking level differences, binaural intelligibility level differences that accrue from having sounds separated from the noise. I would urge you, if you haven't done it, to look at the classic BMLD paradigm in Brian Moore's book, Introduction to the Psychology of Hearing, because he's got a great little picture with happy and sad faces showing the perverse perspective for interaural time difference sensitivity to give you binaural unmasking. You put a noise in one ear and a tone in one ear, in the same ear, and you lower the level of the tone until it's masked. You add an identical copy of the noise at the other ear, and you hear the tone again. Adding noise in that system, which is unusual across all sensory systems, makes the signal perceptible. If you add more sound energy, a tone in both ears now rather than one ear, it becomes less perceptible, and you have to reduce it, have to increase it again to be able to hear it. If you then simply invert the phase of the noise, and this is low pass below, say, 1500 hertz noise, invert the phase of the noise or the tone at one ear relative to the other, it pops out again and you have to reduce it again. So the fact is it's not necessarily an energetic processing system. It's not looking at energy so much as timing, and the exquisite timing is what gets us out of jail in the cocktail party. And we know that if you could put that back into impaired listeners, it would really make a benefit. The other perspective, of course, is that most of the time you're not at a cocktail party. You might be in this sort of situation. And the key perspective of spatial hearing is that not only do you have to know the source or find the source, you also are aware of the room through the ability to process the rapid fluctuating interaural time and interaural intensity or level differences that arise from reflective surfaces and hard objects in the environment. And you feel the spaciness of the room based upon the reverb that hits back at your ears. And of course, and this will come up later, we don't have a situation where we are color coded with respect to the direct source and the reflective sound paths. The red signal is not distinct in sound energy at the ears from the green at the two ears. You just have a single uniform movement of each eardrum. But we know that direct sound paths are, even when the direct to reverberant ratio is negative, when there's more sound energy late arriving, you're much more sensitive to. In fact, you suppress the spatial information about location for the late arriving energy, but it does create or turn into that sound environment. You feel the presence of your living room or your bathroom or an anechoic chamber which doesn't have it, or a cathedral where you have lots of reverb. So you feel the spaciness, but you always have this, and it's known as the Haas effect, as one description of it by Haas, who showed that the ability to have this first arriving wavefront of spatial information dominates not only your perceived location, but also helps you in understanding the speech and background noise. Now, importantly, if you have a room which is very large, reverb helps you understand speech and noise. That long smeary tail, you don't necessarily need to locate. You can see the talker, but you actually can pick up some of the words more easily from that long smeary tail of reverb. So there's a whole important part about this scene analysis that auditory binaural processing really brings to the party. And of course, as I said, it's handling the wet. The difference between dealing with that situation and dealing with that situation is very obvious in your perceived understanding of what the room is. Reverb is critical and likely dominates most of the rooms we're in, except we don't necessarily perceive it as a location. We perceive it as the space we're in. And that's why binaural hearing is critical. We have to square these two factors. Barry Blesser and Linda Ruth Salter wrote Spaces Speak, Are You Listening? Experiencing Oral Architecture. And it's just a sort of a throwaway to say that staying connected to the environment is actually a key part of our listening, and impaired users of hearing aids or cochlear implants often feel disconnected from their environments. There are multiple reasons for that to be the case, but the lack of spatial awareness, whether they have one or two implants or hearing aids, is a significant complaint. And as well as getting a 3 decibel increase in benefit by having another ear, just by having two independent copies of the information, of the energy, you get this connectedness that is all of these features. And losing that connectedness is a major gripe for people who use hearing devices that aren't necessarily providing them with information back about their sound environments, the source location or the room acoustic as a feature. So let's do a bit of history. This is a bit of Blue Skies research back in the 1800s. And there's nothing new under sun. This is Lord Rayleigh, or J.W. Strutt, and this is his house in Turling Place. Rayleigh, of course, won the Nobel Prize for the co-discovery of argon, the noble gas. This is where he did his experiments. I'm not sure why, and he also discovered Rayleigh scattering, why the sky is blue. Although living in Essex, I can't understand why he thought the sky was blue. But he essentially also was intrigued by the long-standing issue associated with how you locate the sources of sound. Now, people weren't stupid back in the day. They knew that for sounds of a particular frequency, there was a head shadowing effect. Your head would be in the way of a sound source for any location off to the left or the right, and that the sound energy would be shadowed at the ear further from the source, and the interaural level difference was therefore a cue to locating the source. They also knew that as the wavelength increased, as the sound frequency went down, the wavelength increased, you simply didn't have a head shadow anymore, or very little, because essentially the wave just passed over the head with no shadowing as the wavelength became bigger than the head, than the size of the head relative to the travel time of the sound. And that meant you couldn't use ILD. So they wondered what it was. And at the time, people said this is impossible. They calculated what those abilities to discriminate a degree or two off the midline were. It was 10 to 20 millionths of a second. And they said, there's no way this is feasible. And it was Lord Rayleigh, actually, who, using one of those classic upper class experimental paradigms, for this experiment, you'll need one stately home, one butler, one wife, two gazebos, two pieces of tubing, and two slightly mistuned hearing tuning forks, demonstrated, and I'll get to that, potentially first demonstrated the ability to extract the interaural phase differences of sounds from the two ears in order to tell the location for low frequency sounds. This is someone else who also claims that, Sylvanus Thompson. It was Irv Hafter who suggested to me in the very Irv way that the upper class nature of Lord Rayleigh relative to Thompson gave Rayleigh the credit and Thompson not the credit for this discovery. I'm slightly not convinced of all of that, but it is well described in Dick's book, and I think you should read the history of this reasonably well. I think there's some, like many ideas, people were coming together at the same time, and it was a useful understanding. What essentially it shows, is just a cartoon, is that when you present a sound with a tuning fork or a sound, let's say on headphones, 1 hertz difference between the two ears, then over time, at 1 second, the difference between the two tuning forks in his case, or tones over headphones in our case, if it's 500 and 499 hertz in the other ear, then you get this appearance of a rotating sound between the two ears. It sets to one ear, flips to the other ear, and comes back. And that binaural beat, as it's known, is pure temporal fine structure information. There's no envelope apart from at the onset of the signal, and you're extracting the phase difference between the two ears in order to get a perception of a sound that is one side or the other of the midline. And that is what Rayleigh had discovered, and that is what probably Sylvanus Thompson also had reported. And this ability shows that you can use timing differences. Small phase differences between the two ears are detectable, and that is a useful cue. And that led to what we now know as the duplex theory, that there are two mechanisms, and this is important, and I'll come to that in a minute, why multiple mechanisms sometimes give you interesting features of spatial listening that aren't necessarily focused on, but do tell you a little bit about how the brain architecture is set up. Here's the acoustic shadowing at 6,000 hertz. For example, there's an acoustic shadow at the ear further from the source. The level difference between the two ears is greater. There's a level difference between the two ears. Your sensitivity to level differences is roughly the same as your sensitivity to absolute level differences. So J&Ds for level and J&Ds for interaural level are about 1 or 2 decibels. It's really for the low frequency sounds where ITD discrimination, the ability to follow 10, 20 microseconds of time difference between the two ears, challenges, because there's no monaural temporal sensitivity that gets close to that. And that's a big challenge to how people were initially understanding how the brain might be set up to explore this. In another Journal Club Classic perspective, and Dick mentions this, Stevens and Newman in the then new building, Harvard Medical Sciences building, and again, for this experiment, you'll need 2 blokes, it's always blokes, with suits, and a range of ways of actually testing in free field without any echoes. So they were on the roof of the Harvard building because they wanted to get rid of any reverberations. And they were testing out your minimum audible angles, your ability to locate, blindfolded if necessary, the location of a source. Obviously, you can see the source here. They were blindfolded for the purposes of being able to discriminate where they heard a source. And Stevens and Newman went through a range of different stimuli and sounds and frequencies of sound in order to map out these 2 binaural mechanisms that might actually allow for spatial listening. This is from their original paper, and I think Dick may have put one of these in here. This is just a couple of their perspectives for the ITD, or for the spatial listening. And what they showed, interestingly, the top figure, figure 2 as they call it, dependence of localization on frequency. And you see that at very low frequencies, you have very low errors in localization. This is an error, absolute error localization, for not for discrimination, just for an absolute judgment. And you can see that as you go up in frequency, particularly above 1,000 hertz, performance gets worse before it comes back again. And this is often the signature of double or two mechanisms that have to be glued together. You see the glitches in performance where there's not enough head shadow, around 2 to 3 kilohertz, to really give you the resolution you see at higher frequencies. We're using ILD, and they're much bigger ILDs, or lower frequencies where you're using ITD. So this glitch is some evidence, as they would suggest, of two binaural mechanisms. The other one that's interesting I'll come up to is the percent of reversals on the figure 4 at the bottom, which shows how many times that a sound which they thought was in front of them was actually behind them. And what you see is that for low frequencies, up again to around this 1 to 2 kilohertz range, there is a high degree of confusing the front from the back with these pure tones. But at higher frequencies, that's not the case, or you at least get much fewer front-back confusions, as they're known. And of course, we know why that's the case. And I use the owl here as an instance, but really we know that there's absolutely no difference in the ITD that's generated by those two sources. So if you only have access to low-frequency ITD information, you can't distinguish these two sources. As you get to higher frequencies, of course, and we'll come to the pinna, there's a much more baffling of the sound energy by the pinna, and that pinna cue is how you resolve front-back confusions. You can also resolve it by moving your head, but the fact is that there are sources, in fact, from a conical surface extending in any size away from your head that yield exactly the same ITD, and it's only the baffling of either head movements or the pinna itself, which can provide cues as to whether a sound is in front of you or behind you, because the pinna has a pinna transfer function, as of course as Simon is well versed on, that helps you disambiguate front from back. ITD alone doesn't, and it's quite interesting that a lot of animals, like birds, that are very good ITD detectors, use pure tone signals. Not in Australia, they're all screeching, squawking, dragon-like sounds, but birds that make very narrowband sounds are very hard to localize, and that's a behavioral mechanism. It basically says all this territory you can hear me in is mine, and it also makes them hard to find, and that's actually potentially an evolutionary benefit for territorial marking. Birdsong is not fun. Birdsong is aggressive territorialism, and pure tones are a good way of actually achieving that. It's also not to be confused with what I call the comb-over confusion. I showed you these two figures before. These are not the same men. They have the classic comb-over hair. One, of course, is Rayleigh. One, of course, is Thompson. And so people do confuse them on the basis of their looks, but also on the basis of who've made the first discovery. I'm with Irv, probably because I'm trying to be a mensch, but definitely Sylvanus Thompson predated Rayleigh's description of some of these things. Also not to be confused, of course, with the cone of silence, which is only for the people of a certain generation, would you believe, where the cone of silence provides a fantastic way of unmasking anything outside and masking anything within the domain. But that also came with the shoe phone, which is now superseded by actual iPhones. So let's move on to a little bit of the physiology. And I mentioned this at the start. I hypothesize, and it's hard to test it, but that the reason we have an architecture that's well known for ITD processing and ILD processing is because it's mapped to the other sensory modalities. Each ear does not have any representation of where a sound object is, or even of the sound object. It's all mixed frequency spectral processing. And a critical feature to get right early on is where a sound is. Actually hearing it, binaural hearing helps you detect things in background noise, having two ears, but also to locate it. And so an architecture that actually is early in the brain, and this is a simplified version of the binaural brainstem pathways. Here you have pathways from the left and right cochlea that go to the dorsal and ventral cochlear nucleus. The dorsal cochlear nucleus decussates across to the other inferior colliculus and the medial nucleus of the trapezoid body. As I said, there's a huge amount of architecture in the auditory pathways that isn't present in any other sensory system. And the one we know most about is the processing for binaural processing. The ventral cochlear nucleus projects into the lateral and medial superior olives, including via some intermediate inhibitory pathways, and has input from both sides. And it's this structure, the medial superior olive for ITD and the lateral superior olive for ILD, that is usually considered to be the breakdown, but I don't think it's as complete as that. And that is where binaural integration occurs. So 3 synapses beyond the ear is the first point at which you can actually extract this information, which means there's a very heavy reliance on the temporal fidelity of ventral cochlear nucleus neurons. Unlike most neurons, these neurons actually improve their temporal processing for certain frequencies of sound below a few hundred hertz. So there's a real capacity of this system not only to preserve the phase-locked information that would come from the depolarization and hyperpolarization phase of hair cells, which are transmitted on the auditory nerve fibers, but also to retain it and maybe even improve it before you get to the medial superior olive, where you have binaural integration that cares about the interaural time difference or the phasic delay between the 2 ears. And so in summary, if you're thinking of the duplex theory, we tend to think of, even for the same signal, if it's processed across cochlear filter bands and it's low frequency at the bottom here, so a narrower filter to high frequency, you extract the information in terms of you've got envelope and fine structure timing differences if the sound is actually heard at one side compared to the other, the red and the black sounds, but only at low frequencies are you able to make use of those fine structure timing differences, and at increasingly higher frequencies, you're left with what we know is the level difference between the 2 ears. The red signal is lower than the black signal at the other ear, but also you have the envelope time difference between these two. It's more apparent in the higher frequencies because the bandwidth of cochlear filters will be wider and there's more envelope at higher frequencies, higher modulation frequencies than you would otherwise get. And I won't talk too much about envelope ITD today, but it's an important characteristic. And then we've always thought about the anatomy being, and this is super simplified, inputs to the medial superior olive from the ventral cochlear nucleus, which are for timing, and excitation from one ear and inhibition via the medial nucleus of the trapezoid body from the other ear, which sign inverts, that actually gives you a push-pull mechanism for ILD, where you have an LSO on both sides. If the sound is closer to one side than the other, this LSO is more active, more driven by excitation and less by inhibition. And if it's on the head shadowed side, it's more by, then it's lesser activation, more driven by inhibition than excitation. And you can see the LSOs on 2 sides of the brain being this sort of broadly tuned push-pull perspective. It's not how ITD processing was considered for very many years. And the other thing we have, and I'll talk a little bit about this, is that, and I think this is very important because it gets around the problem of why people have not been able to replicate or reproduce or introduce true ITD sensitivity, whether in the low frequencies or in high frequencies. By true, I mean very good resolution, the ability to do binaural unmasking, and insensitivity to noise. In other words, low SNR binaural signals are not easily masked because the biophysics of the MSO and the LSO look different. In the MSO, and I'll come to this, and this is part of Jason's PhD work, there are resonant neurons in the medial superior olive act as resonant bandpass electrical filters with a low input impedance, and that means they're good for timing information. Higher frequency cells, probably in the LSO, look like low pass electrical filters with a high input impedance, and they're good for integrating energy, which is exactly what you'd want in a neuron that was determining what the energy at the 2 ears actually was. So the biophysics, which hadn't really been considered to the same degree, is really probably that distinction between being able to use fast temporal processing and being able to use slow integrative energy processing. in binaural hearing. In fact, we can show that, we did show that, and this is part of Jason's thesis, where the intrinsic physiology of MSO and LSO neurons, if you take a brain slice, is critical to the extraction of binaural cues. And without it, you don't get this distinction between level difference and time difference. So this is a piece of guinea pig speech, which looks exactly like human speech. There's all the tracking of the pitch changes, and there's the sound wave there, and you can consider cochlear filters in the system as well. Again, from low to high in terms of spectrogram rather than the other way around. And if you take a brain slice preparation from the medial superior olive and the lateral superior olive, and you hatch clamp these neurons and hold them at a specific potential, you can inject what's called a zap current, which is essentially a rapidly rising or a rising or falling current injection of different frequencies. And what you can return from that is from the same electrode is to record the voltage response. And what you find is that depending on the MSO neuron and the LSO neuron, these neurons are more or less sensitive to a specific electrical frequency input. And that probably drives and their sensitivity to intraoral time differences, and may even be the delay mechanism that we talk about when we talk about intraoral time difference. And MSO neurons tend to show intrinsic resonances. If you put in the signal and you get out the voltage response, you can create from that the impedance profile, and you find that they seem to be tuned for a particular resonance, very fast resonances, faster than anywhere else in the brain electrically. These are very super fast cells. And if you look across the lateral and medial superior olive, you'll see that neurons in the medial superior olive tend to have these low input impedance, very fast resonant electrical properties, whereas neurons in the lateral superior olive, particularly at the high frequency end, tend to have these low pass electrical properties that would be good for integrating information, whereas probably in the low CF, low frequency end of the lateral superior olive, where you have inputs from lower frequencies, and maybe in the parts of the MSO, you have lower frequency resonances that look a bit like a combination of those two filter functions with a relatively moderate but still higher input impedance. And these biophysical features probably determine the extent to which you can actually access temporal fine structure from low and high frequency sounds. Independent of the physical constraints, we think about whether a sound has sufficient wavelength to be ambiguous or not ambiguous for the intraoral phase. And in fact, you can show that essentially by thinking that the resonance, it goes against the tonotopic gradient. The lowest frequency cells have the highest resonances. And as you get to higher frequency neurons across the tonotopic gradient, maybe up into the LSO, you get a transition from differentiating type cells to integrating type cells, which may explain why, and we know this to be the case, either in cochlear implantation or if you low pass filter or high pass filter, should I say, all of your sounds, you lose your binaural abilities. Everything needs to be at positive signal to noise ratio to be able to understand speech and background noise, and you don't have the same resolution for any spatial cues. Low frequency binaural hearing is special. And just to illustrate that, if you were injecting current in the standard way that people do in the lab, injecting current of different current steps into these cells, those in that with a low pass characteristic would show increasing integrative firing for a current injection as you increase the level of the current injection, whereas MSO neurons with their low input impedance only fire eventually, and they only fire once. They fire in these classic brain slice experiments that people would do, particularly in other sensory systems. They are only firing at the onset, and then they're shutting down. They're really not interested in what's going on later on. And that integrated versus differentiated perspective is probably the key characteristic difference between low frequency hearing, which gets you out of jail at the cocktail party, and high frequency hearing, which really isn't specialized for that exquisite temporal processing. Okay, so this model, the Jeffress model, I'm going to explain, and then I'm going to debunk it later on. The classic model for understanding how the spatial information is extracted from sounds which at their 2 ears have no spatial information per se was developed by Lloyd Jeffreys in 1948. And 1948 was a good year for binaural hearing, and Dick's book mentions it. Ira Hirsch and JCR Licklider independently discovered binaural unmasking, the fact that the phase difference between the noise or the signal at the 2 ears allowed you to release from masking so you could hear the signal better. And I think it was often to do with the helicopters and early jet engines that they were trying to get this technical issue sorted out. And simply by inverting the polarity of one of the comms signals in the ear relative to this diffuse identical noise around the 2 ears, the inversion of the phase of the signal at one ear popped the sound out over the headphones of the cockpit pilots. And 1948 in JASA, there are 2 articles, look at them, by Hirsch and by JCR Licklider, actually describing that benefit. In the same year, Jeffreys came out with a model, and this I stole from Tom Yin's lab, I think, and it's been used extensively all over the world to show how the relative time of arrival of sound at the 2 ears is transformed into a neural code for spatial location. And it's going to run fast, and then it's going to run slow, and I'll explain what it means. Here we have the cochlea on both sides. This ball here is the cochlear nucleus, and this simplified input is an array of what are called coincidence detectors, neurons that respond when they get input from at the same time from the 2 ears, innervated by a series of delay lines. These are axonal conduction delays that offset external time differences with an equal and opposite axonal conduction delay in order to generate a place map of the location of the source. So I'll run the movie, if it runs. It'll run quickly and then it'll slow down. So here's a sound source, single wave from one side, generates activation in all of the fibers, but only at neuron number 2 does this single cycle of the wave generate action potentials that converge simultaneously onto cell number 2. And it's the fast temporal and resonant properties of those MSO neurons that allows that cell, number 2, to tell you in this side of the brain that the sound source was in front of you. If you now move the sound source to the side or move your head, you generate an action potential at one ear before you generate it at the other. The same array provides for that location through the fact that although inputs also get to all of the cells, only the coincidence detector where they arrive simultaneously is it sufficient to generate an action potential by reaching threshold and activating. And that is the classic Jeffreys model. It's in 1,000 textbooks. It's been on 1,000 slides. It's probably not the case. But it is a really compelling way of understanding, before there was any physiology, and Jeffreys didn't have access to that, that this was actually what was going on. And it was only, in fact, just after that, people like Mark Rosenzweig and others started recording from the brains of animals for spatial sounds. And I met Mark Rosenzweig actually at Irv Hafter's house once. My first thing was, I thought you were dead, but he wasn't, because it was 1940 something. He actually published something on this. So there is a history to this from the last 75 years or so. But this model is the way the owl and mammals were thought to proceed for quite a long time. I'm going to come back to that in a little minute, but I want to move on to talk about the other element of space that's important, which of course is the vertical dimension and the pinna, which also provides you with externalization perspectives for sound and the dissociation or integration at some point of the horizontal binaural cues and the pinna cues for up down, because they're actually a really important distinction between these two processes, particularly because one of them is particularly well accessed by learning, it seems. This is Alphonse Bertillon, and in the days before fingerprints were understood, he was a detective in Paris, and people use a lot of physiognomy and other dubious methods to characterize the criminal underclasses, including of Paris. So these two characters here, completely identical and indistinguishable, apart from the fact they have different ears. And obviously, that meant there were an awful lot of safe crackers being caught in Paris at the time, because you could get their ear print, put in your ear to the safe. But actually, he stumbled upon something not quite the right way, that we do have unique ears. And the shape of our ears flavors the sound energy that reaches the eardrum, and that is a cue to, and a learned cue to, the location of sources in the vertical dimension. And a great study by Hoffman et al., which is, I think, about nearly 30 years ago now, 25 years ago now, showed, and that was from, what's his name? The lab in the Netherlands, whose name I've forgotten. John van Upstal. John van Upstal done a lot of visual and auditory science, also a punk rocker, and it was in a reasonably well-known band, for a Dutch band. But he basically demonstrated that this map, and Simon is, of course, well-versed in these sorts of perspectives, there is a coding of the elevation for a sound source based upon what your pinna and the concha and all the structure of the outer ear does. And this heat map here shows the gain function of the pinna, measuring a sound outside compared with measuring a sound at the eardrum. And you can see that there are hotspots, particularly at higher frequencies, that seem to be very important for distinguishing where a sound is above or below the horizon, for example. What Hoffman did was to alter this mapping by putting in a piece of this putty. I'm going to call it vulcanized rubber, because you can imagine Star Trek ears having this sort of thing, leaving the ear canal open, but changing the shape of the concha. You can see the transfer function changes. And the question is, what happens when you change the acoustic properties of the outer ear in this way to your ability to locate the sources of sound? And of course, there's our, I've often wondered if we should do a Nature paper sitting there waiting to be written on the localization abilities of prosthetic ears on the Star Trek set. Imagine everyone's banging their head when they had their pinna changed, particularly Mr. Spock. But what they did was to test out for individuals with their normal, and this is back in the day, so they had a screen, they had a black curtain with 16 speakers at canonical positions, which are illustrated by the fine lines. At each of these corners, there is a speaker, and they simply gave people a laser pointer and asked them to point to where they heard the sound source coming from. Now you can see this individual did a fairly good job. There's variance, but they are mapping out the elevation and the left-right extent of the source. The speaker is played. You can't see the speaker. You point to whether you heard it above or below your horizon in that 16 speaker array. When you change your ear shape at day 0, your world collapses to the horizon. You don't have access to your map of your ears. And it's a bit like prism glasses, where you wear glasses that invert the world. The world turns upside down. In this case, the world becomes left-right only. But over days and into weeks, just like wearing prism glasses, you get this, you relearn what's going on. Prism glasses, you relearn the world in the correct dimensions, up or down or left or right. And here you can see that your world expands out. Day 6, day 29, for example, you're back to more or less what you had before. You have this up-down extent. Interestingly, though, unlike wearing prism glasses, you can take off the new ears and you still have your old ears. There's a mapping of your old ears that is retained, instantly available to you, and you now have access to these new ears, which gives the perspective for how you might actually benefit spatial listening in technologies, that you can actually wear better ears than the ones you've got, or you can provide information that's actually useful for elevation that's not actually there on the ear itself, as long as you have the time to learn it. Are we good to keep going? Yep. So I wanted to talk a little bit about where listeners listen in the room. And I showed you this figure before that basically said, you're in a room, and we're not just talking about in the room, but where in the stimulus do you listen? And in terms of extracting ITD information, and how is that possible? Now, I've hinted at that the ITD processing in MSO is very sensitive to the rising energy of a sound. You can do that with, current injection in a brain slice, and you see that the rising input generates spiking, and then the cell doesn't respond, and that LSO neurons tend to encode the energy integration side. So where do listeners actually listen? And can you provide, in fact, what I think is an important element, bringing together all of the elements of sound that have been pushed apart in binaural hearing. We think of ITD in the fine structure, envelope ITD, ILD. We use non-onset stimuli. A lot of ITD processing for binaural studies gets rid of the energy envelope and even de-emphasizes the onset of the stimulus that you have to have before you have a steady state fine structure tone. So we've kind of pulled everything apart. And I want to try and suggest that we need to start putting it back together to understand how the brain actually does the complex listening task of being able to parse sources, backgrounds, noises, room acoustic. Where do listeners listen for spatial information? And as I said, we've ignored the elephants in the room for a while. We've taken a sound. This is actually the sound of an elephant in terms of its waveform of an elephant trumpet. And we've removed the envelope generally when we think about spatial hearing, and we present sounds that are narrowband noises, maybe 400 hertz wide at 500 hertz. 500 hertz is the most expensive frequency ever because every psychoacoustician on binaural hearing tests 500 hertz in there, and they get money to do it from the government, and they present maybe a narrow band of noise, but they tend to not only get rid of the ongoing envelope. In low frequency ITD, the gold standard is to get rid of any envelope difference and to present only a phase difference, an ongoing phase difference. And this is seen as the canonical way of exploring how the brain does processing for ITD. It's very unnaturalistic because there's no ITD in the envelope. There's no envelope modulated information. And in fact, you still do have an envelope. You can't just transition the sound into that. You've got to have an onset. And you've got some ongoing component as well. So how do you get around this problem, which is where psychoacoustics has been for a long time in binaural hearing, and which has really dominated how we understand the binaural and spatial listening? Well, the way we did it, and this is with Matthias Dietz, who's now at Oldenburg. He was at Oldenburg at the time, actually, and then moved to UCL with me for several years and did some great physiology, animal work, and psychoacoustics and MEG. Oops. That's a question, sorry.
Or yes, yeah. You just mentioned 500. I just wondering why 500. 500. Why you use
500 hertz? Yeah, so 500 hertz to 700 hertz are probably the existence region for the best discrimination performance for ITD. So around 5 to 700 hertz is where you get the best thresholds for discriminating ITD. It's where you get the best unmasking of binaural signals. And it's also probably the place where you get the best precedence effect, the ability to extract information from the first arriving wave and ignore the reflections. So there's a dominance region for binaural ITD processing that's around 500 to 700 hertz, and 500 hertz is that standard. Above about 1,000 hertz, you really lose fine structure ITD processing very rapidly.
Thanks.
And really, the question we had when we were looking at this is, well, we go back to Rayleigh's binaural beats. And this has been a question for a long time, because although you can actually discriminate very small time differences and phase differences between the ears, you're very bad at tracking them. In fact, with the 1 hertz beat, you can perceive this rotating tone. If you present it over the two ears, you perceive this rotating tone. But as you go to 2 hertz, or 3 hertz, where each 1 second has 3 rotations, you start to lose the ability to track this. And by 4 hertz, you just hear a blur. You're not able to track changing ITDs to anywhere near the ability that you can track static ITD differences. So being able to track something spatially is very, very slow. It's called binaural sluggishness. And people like John Culling and Quentin Summerfield, Mike Aykroyd, and Tino Trahiotis and Les Bernstein have explored binaural sluggishness in the sense that you can't follow something that's moving. And the minimum audible angle and the minimum audible movement angle are very dependent upon the rate at which you change. The faster something moves, only 3 or 4 hertz, you lose any perception of where the location is, and instead you hear this intracranial, in this case with headphones, image that doesn't move, is static. And it kind of got to us, well, why? How can you do ITDs, but also do spatial listening that really benefits your speech understanding and noise in complex environments, and when things are moving around and sound energy is coming in from different locations all the time. And how can we be sluggish and perform cocktail party listening with these binaural cues? And what we did, this was Matthias's genius idea, was to understand that when you have a 4 hertz binaural beat and you have this broad image, if you modulate the amplitude of the sound at the same 4 hertz, you generate an envelope which is identical in the 2 ears, but now you have an envelope of rising energy, peak energy, and falling energy. And what you find is that becomes a very focused, punctate, and localizable sound image. And in fact, it doesn't occur randomly. The location you would ascribe to this sound, which has a binaural beat at 4 hertz, and every cycle is amplitude modulated, so that each cycle of 360 degrees phase difference is encompassed in a full amplitude modulation cycle. What you perceive is a sound image that is off to the side, where the red ear is leading the blue ear in the energy rise in this case. The location of this sound source is always perceived just where the energy is rising out of the dip. You always perceive it, in this case, off to the right, even though the peak energy might not say that. In fact, we did this systematically and showed that in this case, when you hear a sound where you can change the phase, starting phase of the signal within the envelope, you can see that the peak energy is on the leading on the left, on the right ear. The red is leading the blue, but you actually perceive the sound off to the nearer the left, because the peak energy is just, so the rising energy is in this phase is just leading at the left, even though it's very quiet energy. And we've systematically explored this and found that whenever there's an energy increase, you extract information from the rising energy just out of the dip, which makes sense because the first thing to hit you as sound energy will be the source. Everything else that comes later is likely reflections. And so it makes sense to have a system in the medial superior olive, we predict, that picks up the rising energy and shuts down for localization information. And in fact, we can see this. We went on to show this in medial superior olive and inferior colliculus neurons in small animals that shows the same thing. Neurons at that part of the brain actually are already only responsive to rising sound energy, to the ITDs conveyed there. Even though the ITDs are right through the whole thing, when you put on a rising energy condition, they extract information from the rising envelope energy and they stop firing. And we did MEG for this for neural correlates of perception and found that, in fact, and we'd done this first before we did the animal work, that sounds that have the same perceptual location, independent of how you get there, are essentially perceived as the same, and your brain processes them as the same. We used an adapter paradigm to demonstrate this. And in this case, we had the same sounds. We had an adapter, which was a static IPD of 0 IPD. So we have an envelope at 8, 16, or 32 hertz. We used, in fact, 16 hertz envelope and a 16 hertz binaural beat. And the ITD is 0 the whole time. We then have the parameterized, what we call amplitude modulated binaural beat, AMBB, where we can change them what the ITD is at the modulation minimum. In this case, it's also 0, but you can see because there's a binaural beat, there's a 8 hertz frequency difference between the 2 ears. The fine structure drifts from leading at one ear to leading at the other over the course of each cycle. And the question is, what does your brain cortical activation look like to these two sounds? If they were similarly perceived, we would predict that they adapt each other out, because we're going to present these continuously, 500 milliseconds of one, 500 milliseconds of the other for 4 minutes, and record using MEG your auditory cortical responses to these sounds. And what you find is that when you have an adapter as a static ITD, in this case of 45 degrees interval phase difference, negative 45, leading on the left, and an amplitude modulated binaural beat with a modulation minimum of 0, that's the adapter and the probe going on, then you find that, in this case, sorry, this is 0 again, you find that that one with a 0 ITD and a modulation minimum at 0 ITD activate in the cortex fairly high responses. And they're not, and that's because we say they're not perceived as the same. If you take the same probe and put it with an adapter where the static IPD is 90 degrees, 90 degrees IPD for the static would be somewhere up here. For the modulated binaural beat, the IPD in this stimulus at 90 is about there on the rising energy. And what you see is you don't get a response. They're essentially the same and they adapt each other out. You perceive them, but your neural correlate at some level says these are the same signal. Even though they're not the same signal, perceptually, most of this signal is driven by that ITD. If you then put the energy, the IPD, static IPD that will be consistent with a falling energy, so the IPD of the adapter is consistent with an IPD on the amplitude modulated binaural beat over here, where you can see the blue is leading the red, you get a response again. So energy determines the extent to which you can extract ITD. And that is a critical feature that we think is missing in the way people explore ITD in the temporal fine structure. That the envelope and the fine structure temporal information together is important, and you can't actually separate these two things out. And of course, the envelope ITD information is also going to have some sort of level difference between the two ears in it as well. Just very briefly to show you this, and then I will finish up. In fact, we now think that you... Oh, question, sorry.
So for this kind of experiment, do you have to choose the subject? How are you going to choose which one you want to do the testing on?
Well, anyone with normal hearing, which is important. So you have to have normal hearing listeners with abilities to do ITD processing, so normal binaural hearing. And those subjects were just chosen. We did that imaging in Finland with our colleague, Nelly Salmonen, when they had the MEG system there. And there was really very strong consensus across all listeners. All listeners were picking up the energy. Our hypothesis is that people with impaired low-frequency hearing or no access to low-frequency hearing, like a cochlear implant user, won't be able to show this. And in fact, if peak energy is associated with reflections and late arriving localization information, maybe peak energy discrimination or peak energy models for cochlear implant stimulation are not the best when it comes to spatial listening information. It's the rising energy out of quiet that is the most consistent and reliable ITD information.
So when you
sorry,
when you perform this kind of study, what's the sample size you want to use?
Oh, we had about 14 listeners in that case. Yep, so it was a reasonable amount. So 14 listeners who did the psychoacoustics and the brain imaging.
Thanks.
What we then found in the lab really is that listeners are paradoxically or maybe counterintuitively or even just counter the received wisdom are actually insensitive to ongoing phasic ITDs. If there's no envelope, they can't extract the information from fine structure, which is perverse. But here we have a situation in which we have two envelopes of sound, and it's a low frequency centered noise, bandwidth of about 300 to 700 hertz, which is the standard stimulus centered at 500 hertz that has been used for psychophysics for 50 years. And what we do is we do a non-zero ITD in different segments of this envelope, either this fast attack, fast fall flat envelope, or this slowly rising SAM envelope. In either way, we put the ITD somewhere at the start, middle, or end of the stimulus, and the rest of the ITDs are zero. And the question is, how well can you actually tell two sounds as to whether they have an ITD in them or not? And what you're doing is presenting a sound with an ITD leading on the left and one leading on the right, and what's the smallest ITD you can discriminate for a sound that is the first signal in a trial will have an ITD leading on the left, the second signal in the trial will have an ITD leading on the right, and what's your minimum discrimination performance for that? And the ITD only, the non-zero ITD only occurs in one of these segments. which sound was on the left. It's a very simple experiment with a staircase adaptive process. And what we found, and this is for all listeners versus our best 12 listeners, was that in fact, the envelope shape completely dominates your performance. The abrupt envelope, which is the red data here, where you have a rising energy and then a very flat sustained perspective, shows, that's the red one, thresholds of about 100 microseconds if you put the ITD at the onset. But as you put the ITD at the energy peak, performance really goes up through these are very poor resolution ITDs. You can't tell left from right. In fact, not only can you not tell left from right, if you look at this figure over on the right here, these are the number of trials they couldn't do the task at all beyond 1,000 microseconds. 1,000 microseconds and they were out. They were tracking this. They just couldn't do the task. 60% of the time, when the ITD is in the energy peak, people were unable to actually achieve a threshold for a plus or minus 500 microsecond ITD, 1,000 microsecond difference. When the energy came down again, was falling again, they got back to normal performance. With the slow envelope, you see the performance stays high, or good, should I say low, thresholds are low, when there's any possibility of rising envelope, even if it's a tiny bit. But the minute you start getting slow changes in energy on the way down, performance goes to the roof, and you can see that in the group performance. Pretty much 100% of people couldn't do the task. So the energy envelope is the key determinant of how you can or cannot extract ITD information. And the rising energy is the determinant feature, and the speed at which it rises or falls is actually the key features in extracting information. And of course, in a room where there's lots of reverb, there's going to be slower, lower frequency modulations, etc. You're going to have to rely on very rapid changes in energy. And Jason has done some excellent work on this with Andy Bruguera to demonstrate why that's the case in natural listening without reverb and natural listening with reverb, for example. You have to use onset ITD information. So the moral of the story is no envelope, then you know your elephant. No envelope, no elephant. You really do need to have, and this is important because the energy envelope tells you about the source. It tells you what it is. Is it male, female? Is it an elephant? Is it deers and the vocal tract length? It's a measure of the vocal tract length. Is it fit for mating with or not? And you can't delay the location from the object. And putting them together in the same space and doing it very early in the system, not waiting until you get to the cortex, is important. Knowing your envelope and knowing your elephant are the same thing, because you need to know the source, you need to know the room, you need to know what the object was. You can't just run with temporal fine structure and then some lazy part of your brain later add it in. It's got to be with the object, and that object is often driven by the knowledge of what a vocalization is, male, female, species, etc. Can I have 2 more minutes and then we'll finish?
2.
Yep. Okay. Just want to say your binaural brain is smarter than, oh, Dick, sorry.
Yeah, before you go on, there's a thing I mentioned, I think somewhere in the chapter, that there's been speculation that some of the adaptive mechanisms in the cochlea and in the hair cells will have enough onset emphasis to sort of explain the precedence effect, that they'll give you a much bigger response when the envelope is rising than when it's falling.
Yep. And that's absolutely true.
Yeah. Have you looked at whether that's enough, or do you need still more specialized mechanisms in the?
No, you need only that. In fact, adaptation before binaural integration is necessary and sufficient to account for the performance. So when we originally did the study with MEG, we did the MEG and behavioral performance before we went to the small animals. We went to the small animals and looked and saw that and we predicted that you'd need monaural adaptation before binaural from the nerve. And that is indeed the case. You can account for the performance by that early cochlear nerve fiber adaptation and possibly even hair cell adaptation. And Jason has significant expertise in that. I just want to talk about when there isn't an envelope and what an envelope actually is. Your binaural brain is smarter than you are, because you know that if you present 2 pieces of noise that are independent at the 2 ears, if you listen monaurally, you can't tell the noises apart. They're the same long-term statistics. It's very hard to tell them apart. But if you hear them as a sound at the 2 ears that is independent noise at the 2 ears over headphones, you hear a fuzzy inchoate sound, and you can't move it around by imposing an ITD because the temporal fine structure is independent in the 2 ears. It's random. And this is known as the interaural coherence, the extent to which the sound of the 2 ears is the same. And when it's not the same, you get this broad intracranial image which doesn't move around if you impose an ITD. If you put the same noise on, then you get a focused punctate sound image. And that ability to have a compact versus a compact sound image compared with an inchoate one is probably based, has a whole range of things that we're interested in at the moment about the statistics of the environment, certainty with whether sources are separable. If a sound has a big image, it's less separable from others. And of course, this is the feature of interaural coherence between the ears that tells you about the room acoustic. If sounds are getting interfering with the walls and other hard sources, you get a less coherent sound image. The room sounds bigger and sounds more reverby. Now, if anyone had a set of headphones, I was going to put this sound on because this sound, you can play it later, shows you what you can do with ITD. In this case, if we present a sound with non-identical at the 2 ears, this one's identical, but in very short periods, we transition from incoherent to coherent over time in select frequency bands, you can generate not just a perceptive sound, you can generate a melody with that sounds like a musical sequence. In fact, this sound, if you listen to it, I'll play it. If you're listening to it without headphones, binaural, all you hear is noise. If you listen to it on one headphone or the other, all you hear is noise. Listen to it together and you hear structure in this sound because we're transitioning to high coherence over time in different frequency bands. In fact, what you'll listen to is Somewhere Over the Rainbow. You'll be able to hear Somewhere Over the Rainbow being played in that sound because you're transitioning very briefly into oral coherence. There's no change in energy at the 2 ears. You're insensitive to it at the 2 ears all the way up to the MSO postsynaptically. But the transition to coherence is sufficient to generate the percept of an auditory object that you can follow as a melody. And it really challenges what we think about what envelopes are, because it's an internal only envelope. And you've generated what I think is and people know these dichotic pitches, Huggins pitch, binaural edge pitch, things like that. But in fact, what it's telling you is that the energy that you can extract from coherence, especially in the complex listening environment, is probably what gets you out of jail free. These small transitions in energy that in coherence that have no energetic difference, but are small transitions to highly coherent between the 2 ears in ITD and ILD is sufficient to generate objects. And that's probably how we do our binaural listening. And I'm going to end by saying that when we measure these objects as compact or diffuse in the brain, what we find is that very compact objects, so the coherence is high compared with the interaural coherence is low, very compact objects generate very little activity. Inchoate, uncertain, diffuse sounds generate a lot of activity in the brain using the functional near infrared, which measures the blood oxygenation. It's a proxy for energy use. And so to some degree, the purpose of our binaural brain and those things is to reduce the amount of energy by having highly compact, sparsely represented images. And the experiment that we're writing up for a grant now that Havett and I are writing up is essentially to explore this sort of feature, uncertainty. Can you actually put objects into this with this method I showed you and actually reduce the amount of brain activity when an object appears because it's a certain object? Whereas inchoate, diffuse listening environments are complicated to deal with and every possible vector is there. You can't distill it. When an object appears, certainty increases, energy goes down. And that's, I think, with the launching off space for our next understanding of spatial hearing, moving it into that prediction, development, learning phase, and into that energy and efficient use. And the last few slides I'm not going to go through except to say this is the journey I went on for 25 years. 25 years ago, the Jeffress model was about 50 years old. And it took me a lot of papers and these are just the showy ones, over the last 25 years, including this one published last 2 weeks ago with a bunch of us on it, including Jess, to show that the Jeffress model is not correct. And that what we see in small animals, and I won't go through all that, it's another different story, is actually what we see in humans. And that the representation of ITD is not like the Jeffress model, but is a more dynamic, abstract representation, which scales according to efficiency, head size, frequency range, and is adjustable over time, and gives you simultaneously location, spaciness, reverb, coherence, all with one very small set of neurons. There are maybe 100 or 200 neurons in the medial superior olive. And that is what we get for being able to extract all of that information at the same time. So that's now a 75-year-old model, and I'm no longer 20-something, I'm 50-something. But dealing with that model and showing that it's probably not true, even though it's elegant and beautiful, I think isn't just an exercise in science. It's a demonstration that there are new ways of thinking about the technologies we might develop from these more efficient and dynamic spatial coding mechanisms. Thank you.
Great. Thanks, David. That was great. I might be a little biased, but that was a lot of fun. And I think you've made a wonderful segue into, potentially a follow-up lecture in the not too distant future. But let me open it up for questions. I'm sure there are plenty. There was plenty appearing in the chat, so maybe people want to reiterate some of the things they put in the chat.
No, everyone wants coffee.
Drinking from a fire hose, mate. That's what it's like. David.
Thank you so much for a wonderful presentation. So many interesting things to think about. One of the things that I was wondering throughout, often we have to gather a lot of information to discern certain kinds of effects, and there's a lot of averaging that goes on. There's a lot of accumulation so that we can discriminate signal from noise or systematic effect from randomness in what we're measuring. So just in broad terms, how much averaging or gathering of information over let's say periods of time in individuals or across multiple individuals, how much of that has to take place for us to start to discern the phenomena that you've been describing here? The things stand out clear as a bell right from the get-go, or is it something where we really have to gather a lot of observations to discern things?
Strangely, it's been the opposite, and yet it's the wrong way. So if you look at the psychoacoustics papers in JASA, for example, it'll be 4 subjects who are all highly trained and selected for being able to do the task. And then the whole task has been focused around ability to discriminate ITDs or envelope ITDs only. And unfortunately, that stripped out the one factor we know is critical across humanity, which is variance. I had this discussion with Bill Hartman, the last ARO I went to before COVID, where I was actually making the point that variance across listeners in these tasks explains what's going on in the brain. And often what we see is that the great variability they might have for threshold performance, for example, if you don't self-select, I mean, often in psychoacoustics, it's myself plus 2 other people with a vested interest in this paper being published. But even when it's selected groups of people, they're selected on the basis that they do the task better than anyone else. What we find when we look at the variance associated with a performance in a task, there's another dimension across which the variance is explained by that. A good example for us, and we haven't published this and Jess won't like me saying it, but we looked at the ability to do envelope ITDs, so just ITDs conveyed in high frequency envelopes, no low frequency fine structure in there, just high frequency sounds with envelope ITD imposed. And Jess was, and a couple of other people, including her twin sister, which may not be irrelevant, were phenomenal at being able to extract envelope ITDs at a 6 kilohertz carrier with a 400 or 600 hertz modulator, which is information and temporal and modulation must be down on the skirts of the filters by that stage, yet they could get 40, 50 microsecond ITD. Other people, Jason being one maybe, myself being another, were way at the other end, like couldn't do the task. And yet, when you then looked at things like speech and noise processing, there's a dimensionality that's opposite to that for the same listeners. Matthias Dietz in his thesis was the outlier. At 1,000 hertz, when he compared ITD in the fine structure opposed with an ITD in the envelope, you can follow one or the other, but one would tell you you're on the left, one would tell you you're on the right. He followed the envelope ITD, and the 3 other subjects in his JASA paper followed the fine structure. And so a sound that's identical is heard on the left or on the right, depending upon you. And that you is probably your biophysical constraints. He was also heavily interoral level difference dominated at 1,000 hertz, and they weren't, which says that he is an integrative brain and they have a differentiated brain. But I mean, it's a difference between, that's the physical dimensions of the universe, left, right. So we've missed the variance, and missing the variance stops us understanding why people do or don't perform. And it's something that David and I are going to be looking at, including with Sherm Lingham, including, what's the variance of listeners? The variation in listening performance is probably understandable if we look at these features that are maybe down to the biophysics. We know your heart and the muscles you have in your leg, and whether you're a fast runner or a slow runner, can depend on potassium channels. Your spatial listening, whether something sounds on the left or the right, probably depends on potassium channels. And that's a big distinction between the way the signal processing view, that it can all be done by the acoustics, and this variation. So I think we have to expand the range of people we look at. And this is something we do, and use defined groups. Autistic people are a good example, who have hypersensitivity and hyposensitivity. How's that? Well, it may be to do with their ability, for example, to extract information that we ignore by integrating it into longer term, and they're able to pick out and are maybe annoyed by very quiet sounds or very snippets of sound energy coming from places that we are integrating, and they'll be able to do certain psychoacoustic tasks better than us, but less able to cope with room reverb, which is quite common.
Well, as the French would say, vive la différence. I think
Vive la
it's probably
différence.
good for us as a species,
Exactly.
Yeah, great.
David, thanks so much. I must say this was perhaps the funnest chapter to write, because of such colorful history and so many interesting theories and personalities and so on. But one thing that struck me is that there was way too much reliance on pure tones, and in more recent times on Gaussian bandpass noises. Correct. Which have kind of the least interesting stuff in their envelopes, and they're about as far as you can get from what we really localize. So, yep. Like speech has these really nice onsets with every pitch pulse.
Yep.
And the distributions are very long-tailed, unlike Gaussian noises. So do you think experiments with better signals like that are gonna be helpful in elucidating the mechanisms even further? Or do you think the kind of impoverished stimulus helps you get at things better?
I think the issue with the impoverished stimuli is that the models then became independent lines of evidence around those, and it was very hard to move away into what we know. We know binaural unmasking is an important thing. We know speech and noise, spatial release from masking are important things. Cochlear implant user doesn't really care if they can do microsecond ITD sensory. They want to know, can they hear someone talking and understand the conversation and know they were on the left, for example. But the two camps have stayed separate. Our job in 25 years of this has been to pay homage, and not just homage, be faithful to the psychoacoustic paradigms because we had to bring those people with us. I mean, we always use the 500 hertz center frequency with a bandpass noise of 400 hertz wide. Not always, but often, simply because we have to be able to link back to that into real world listening. Some people just ignore it. Josh McDermott has really understood that some of the people aren't co-optable, and he's done this cool way of, naturalistic textures and things. I think it's a really good way of doing it. But the hardcore, and I would say the technical hardcore, has been down in those psychoacoustic paradigms, and we've been careful with our big showy papers to bring those people with us. Sometimes you can't. You've got to avoid them in review. But we use the stimulus that demonstrates that your stimulus says this. And some of those papers, our very first fMRI paper in the midbrain we did with Tim Griffiths back in 2005, used exactly the same 500 microsecond ITD on the left or the right, 1500 microsecond on the left or the right, which is beyond our pi limit, and you can read about that, and showed that there are no detectors outside this pi, which is roughly the size of the head, around 700 or 800 hertz. There's no detectors. There's no explicit representation of ITD a la Jeffress, and that you have to make use of the phasic sparse representation you have to do any task. But those are difficult things to get across when there are built models that really do rely on those. And they have tended to ignore what I think is the really important part of binaural hearing, was the real listeners, real variance, real need, and the speech being that key part of it, in my view.
Tim.
Hey, David, great lecture. That was awesome. Thanks.
Thank you.
I am, of course, going to ask about cochlear implants.
Yep.
And maybe just to simplify it all, we won't go into bimodal, just bilateral cochlear implants. Given the 25 years of research on this, what would you do in a bilateral CI system to improve binaural hearing in CI?
Well, I mean, personally, I think you guys have been on the right track for a little while, but it's hard to initiate. Getting apical stimulation effective is the challenge. And I think getting good apical stimulation that gets you into those biophysical cells, and I've said this for 25 years, and I remember saying this at I think it might have been a cochlear conference or a cochlear workshop at a conference with Jan in the audience and others years and years ago when I was still in the UK. And this isn't the grey wobbly jelly. There are hardcore biophysical principles going on in the different cells across the tonotopic gradient that do or do not allow you to pass that information. And if you can't get it in the right channel, that's the first thing. So if you can't stimulate the channel, it's complicated. You then have all the things that Dick talked about. You need adaptation, you need adapting firing, and you need it to be effective. But I guess the other thing is when you look at people who are trying to get binaural fusion going, which I think is the first stage for having more certain spatial listening, a more compact sound image, they talk about changing the ITD, if it's electrical ITD or whatever, to get people to report a more compact binaural image. But the paper I'm just showing you that we just published shows that binaural image compactness is not linearly associated with ITD. It's phasic. It's not phasic on the period of the stimulus, although it's close. It's phasic based upon the neural distribution, the stimulus, and the head size, because you can do it in macaques and humans. And people's perception of the spaciness of a sound as you change the time delay goes up and down. So it might be in a binaural fusion matching, you get a local maximum of fusion, but it's not actually the global maximum of fusion. And we know that impacts performance. Long ITDs are less discriminable, and binaural masking is worse at long ITDs. Even if you're in the right phasic relationship, you still have a local performance change, but the performance changes like that as you change the ITD. So changing something you don't know, which is what's the actual effective time difference between the 2 ears in a cochlear implant bilaterally, and using fusion is a good idea, but you may only be finding the local fusion maximum, not a global fusion maximum. And that will affect listening performance. That's maybe subtle but complex, but useful.
Yeah, definitely.
I want to jump in and grab the last couple of minutes before we run out of time and people drop off. I want to thank David and all the speakers we've had in this series. I think it's been incredibly illustrative, and I think we've created an absolutely wonderful resource for the community. I want to thank Dick for actually writing the book and bringing together all this material into one space, which has been real sort of fuel to the fire here. And also I want to thank Dick for being a wonderful provocateur during the presentations as well. I think it was you added an awful lot of value there, mate, in terms of focusing on things and really drawing out the important threads. We had floated the idea of maybe continuing this series. I sent around a Word doc to collect some ideas. I'll send that around again to this list. We have a few ideas on that, but we haven't got anything planned for next week. So when I send the doc around again, please add things that you would like us to, organise some talks around, and I'm happy to put some time in and do that. So watch this space. And although we'll probably take a little break over the next week or two, I'm very hopeful that we'll be back with some follow-up lectures. We might even start with David continuing his arguments about head size, etc. But anyway, maybe we'll need a little relief for that, and 2 weeks won't be enough. But please look out for notices and notification of other lectures, because it's going to be fabulous to, I think, keep this initiative rolling to some extent.
Yep, agreed. And the characters are no less characterful. Well, they may not be a smart dick, but the binaural world still has a bunch of characters, of which some of us can testify personally.
Certainly has. I know some. Yeah.
Present company, except of course. Okay.
All right.
Thank you all.
Thank you,
See you all.
everyone.
Thanks, everyone.
Oh, very much.
Bye.
Thanks. I'll miss you.